all that being the 8, I think it was.
Yes, so I just started the recording. So yes, I will repeat that.
Thank you.
So yeah, so the foundation are the 3-2-1, which is the total gut restoration, 3 bottles of Megaspore, 2 of the Mega Pre, and 1 of the Mega Mucosa. When you have individuals that have a very damaged immune system and they have recognition issues, this is a Treg problem, then they will better tolerate starting slower with fewer spores. So the 1 spore probiotic is called HU58, and then you would graduate them to the 2 spore probiotic, which is Restore Flora, and then you graduate them to the 5 spore probiotic of Megaspore. And another huge help will be containing the toxicity that is released from those pathogens that do not want to be evicted from their home. They release lipopolysaccharide, which is in the cell wall of many of these organisms, and you can shut that down with IgG 2000. So this product, Mega IgG 2000, are concentrated immunoglobulins that will attach to viruses, parasites, bacteria, lipopolysaccharide, C. diff, H. pylori. There is a whole list of antigens that this product is very successful at neutralizing. So many times when you start with Megaspore, this is like a diagnostic tool in itself. It will give you a very good idea of how much dysbiosis, how much barrier damage, and how much immune dysregulation is going on. You start them at 1 capsule every other day for a week, then 1 capsule a day for a week, and then by the 3rd week, you're up to the recommended 2 capsules a day. You're giving these with food. But when you're starting out, if that 1 capsule causes any GI stress, and GI stress can be as unique as the fingerprint of their microbiome, and it can include gas, bloating, cramping, constipation, diarrhea, hives, rashes, foggy thinking, depression, anxiety. Hopefully not more than 2 at a time, but anything is possible when you're dealing with individuals. So what you tell them is you can always slow down. You can always open the capsule. You can always use a 1/2 a cap, a 1/4 of a cap, an 1/8 of a cap, or pinch of a cap and sprinkle it on your food. But if all fails and they absolutely cannot tolerate Megaspore, then you do that stair-step protocol that we discussed. If you're dealing with someone you already know, everything you have ever tried them on, they could only use a fraction of it, then assume that they're hypersensitive, pre-treat their gut with the IgG 2000, and then start introducing the stair-step sensitivity protocol. And that way you're identifying the 20% of individuals that don't tolerate Megaspore up front, so that these 20% don't take up 80% of your time. Okay, so that's really the first thing you're looking for, is who can tolerate, who cannot, and then you're looking to see if there's a necessary need for any of the other products. If you have someone with GERD or SIBO, then you can assume they have, a pH barrier that it has been destroyed. And this is a very good upper digestive aid. It's helping with GERD and dyspepsia, H. pylori, and what we call phase 2.5 detoxification, which is helping the bile to completely remove toxins from the body. You'll see that many SIBO issues are made worse because the pH barriers are not intact and the lower gut organisms are translocating up the intestinal tract. And if you have sluggish bile flow, that bile can work as an antimicrobial, but if it's not being produced, you're going to have more overgrowth in the upper intestinal tract. So this is a great product for helping the bile, helping the pH barriers, and a lot of that upper digestive issue. The Omega is a very concentrated anti-inflammatory, for the gut. And we raise the bar on omega fish oils because many of them don't quantitate the pre-resolving mediators that are the primary anti-inflammatory molecules. So that's a great product. The Wheat Rescue is really focusing on the inflammatory aspects of gluten and gliadin. So this is going to help in your education of your individuals how to reduce the inflammation in their diet and their lifestyle. Mega Omega is going to be back ordered for a while. I don't think they have any more in stock. It's a fabulous product, but unfortunately, the gel cap that it's in, the product was so strong that it dissolved the gel cap. So we're reformulating that. These two products, besides Megaspore and the spores, are my favorite because the number of vitamin K2 dependent receptors in the body's tissues, glands, and organs will amaze you. And a lot of the issues that we're seeing today is because calcium is depositing in soft tissue where it should not be. And our diet is not sufficient in vitamin K2. The microbiome that is supposed to ferment vitamin K2 is not working properly. And the overuse of vitamin D3 is causing a perfect storm where individuals, their soft tissue is more rigid. It's their heart rates or their blood pressures are going up. They're forming more kidney stones and gallbladder stones. They're aging faster because the elasticity in their tissue is more rigid. And all of those can be helped with megaquinone. So I start them on this formula first because it has some magnesium, zinc, and a little vitamin K, and fill up their nutrition buckets. After about 3 months, I transition them to MildMax, and then you won't be doubling up on magnesium and zinc. So that's kind of how I view the product line. I see how far I can go with the total gut restoration, and then I fill in the gaps with the rest. The most recent products are these immune products. And these are great, again, for all the reasons I said about COVID. And if you've got high gut inflammation, you're going to have more comorbidities and you're going to have more problems with the virus or the vaccine. And so, we can go that direction. We can go more into the direction of how the mechanisms work within the total gut restoration, or we can talk about some specific protocols you have in mind. Just let me know. Or we can talk about the BiomeFX, which is the stool kit, which is really revolutionary for the stool testing industry. So let me know what next step you'd like to do.
Okay. I'm curious about how, so from what I understand so far, is that you're offering testing, I'm guessing it's through that stool kit, where you're able to see, based on the results of that test, where someone's gut bacteria is already. And then I understood everything you just said about the general overview of the products, but would the supplementation of your products happen based on the results of the stool sample?
Okay, so the strategy for the test, most of our practitioners are going to do the stool test and then immediately start their client on the total gut restoration. Then in 5 weeks, when they get the stool test results back, they're going to be able to fine tune the protocol, knowing more about what's going on in the community of bacteria, but having already started the repair process. So they're that much further along by the time they get the results, and then they fine tune it when they can see specific pathogens, specific ratios of keystone strains, and be able to educate better on the lifestyle, supplements, and diet recommendations. Did that answer that question?
Okay. It did, yes.
Would you like to go through the report summary to get an idea or an overview? I don't know how familiar you are with stool testing.
Not familiar with stool testing. I was trained in neurotransmitter methylation, metabolic, and sex and stress hormones as far as labs go, but not stool tests. So this is new.
Okay, we'll know that you haven't missed anything because prior to our test, the analogy is you were looking at someone's ear and trying to identify them, where we show you their complete face. So that's really the difference in the technology jump that we have made by introducing whole genomic sequencing. All of the competitors have a technology called 16S, and they use PCR. So they use 5 to 9 amplificons, and then they amplify it so they have an accuracy that's only 30 to 50%. Ours is 95 to 99. So big difference there. Okay. So if you were to look at this summary, the first page is very helpful.
Can I ask a question?
Yes.
Okay. So I mentioned you said something about the genome being involved in the testing, and I'm suspicious about any kind of genetic testing because I know I have severe distrust for big pharma, and I know they're doing a lot of DNA harvesting from people right now in terms of the vaccine program. And I don't want to sound too conspiracy theorist, but I have doubts about that. And I'm wondering if any of the big pharmaceutical labs are involved in how you process the results, or if this is independent from that medical system.
We are not associated with any big pharma companies, and I have the same concerns as you, and we can talk more about that when I stop the recording.
Okay. Okay, I see.
Okay, so the first page, you'll find that once someone does any test, as soon as they put it in the mailbox, they cannot wait until to get these results back. And when you do get the results back, by then they're already worked up, wondering, is it going to be good? Is it going to be bad? Are they going to survive? And this is nice to have an overview so that you can immediately put their mind at rest. This color graph is showing, if we want them in the green, that's the best. In the yellow, obviously there's some optimization that can be done, but red is going to be a problem. So they can see right away, okay, I'm not terrible. There's some things I can work on, and already they start to relax. So, this is very helpful. And then you break it down. What are those things that can be optimized? And the first area to look at, of course, is diversity. We know that the most diverse diet is going to foster a more diverse microbiome. So we look at alpha diversity, which is saying, do they have the right gut bugs? And then beta diversity is, with the gut bugs they have, are they well adapted to their diet and their geographic area where they live? We do samples from all over the world, and you can drastically see by the beta diversity if someone recently moved here from Africa, for instance. They might have good gut bugs, but the gut bugs aren't adapting well because they're a recent immigrant or visitor. The other area to look at is antimicrobial resistance. We have to understand that the community of organisms within our gut, they have friends and they have foes. They produce metabolites for their friends and bullets for their foes. Those bullets are in the form of defensins, antibiotics, toxins. And if they're getting along well, then they're going to have more friends. But in this particular sample, we see that they're not getting along very well. We want them in the green. They're down here approaching the pink zone. And what that's saying is that this individual is going to be vulnerable. their gut has turned on themselves. And if they were to be exposed to a virus, for instance, their resistance would be down. So this gives us an idea of how protected this person is. And then we want to look, well, why aren't they being well protected? Could it be a pathogen? So we do look at the most common pathogens. And here you see they're not doing all that bad. So it's really not a pathogen that is the problem. They have one that's being detected. And, you know, if we look into what the biochemistry of this particular organism, this organism likes to grow when the bile is not working well. So it's associated with bile, biophilia. The name is a hint for that. But there's only one, and it's not doing too bad. Then the next area to look at are in the commensal organisms.
Can I ask you one question?
Yes.
Okay. Can you repeat what you said about the biophilia? Is that a strain or what? I'm sorry. Just repeat that one time for me.
So Biophilia
What's happening with that?
wadsworthia is a bacteria, and it is considered a pathogen. And just because pathogens are present doesn't always mean they're causing a problem. This one is considered high, so it is out of range, and it is causing a problem. And most likely the problem is associated to poor bile flow. And that's the hint of what this organism's pathonicity is in its name, biophilia. Okay, the next module are the commensals. And out of the commensals, there are some that are more important than others. And the more important ones are considered keystone species. That means the research that's been done, these organisms are associated with more optimal health than other commensals. You can think of the keystones like the keystone cops, that these are the ones that keep the peace and keep things in balance. And if they're out of balance, either too high or too low, then they're trying to compensate for a unhospitable condition. And we already know that from the AMR, there is some hostility going on in this person's microbiome. And one of the primary things the keystones are in charge of are producing metabolites. And the metabolites are part of those functions that we're seeing listed here. These commensal strains produce the very important short chain fatty acids that drive the mitochondria of both the good gut bacteria and of the host, being us. They're in charge of neurotransmitters. The bacteria in our body have 100 times more genetic material than the human cells of our body, and we depend on them for the biochemistry to function as a human. So these bacteria produce neurotransmitters, they produce vitamins, they produce short chain fatty acids. So at this, for the first page gives us a lot of information. At this point, we could go right, to the recommendations and start giving a protocol. If that's the only page that we looked at, we would be able to do that. But if someone is nerdy, like a lot of my friends, we go deeper into this report to find out more information. And you'll see that these reports, if you were to read any of the publications on metabolomics, on microbiome, that this is the latest science. These are the scattergrams and the graphs that are being published. You won't find other stool testing technologies used, because they're using the whole genomic sequencing in publications, in government agencies, in universities. So, this is the latest technology. Now, this is an interesting diagram in that it would be like looking down someone's throat, and if all the organisms actually had different colors, you would be able to tell what was going on. So this is the healthy population representation, and we're looking at the phylum level. So here, for an example, you could say that this is looking at how many humans to how many chimpanzees are in your gut. And if you look at your sample, well, you've got more chimpanzees than you have humans. So that's a problem. And if you look at what those colors represent, you know, they have the Firmicutes versus Bacteroides, for example. And we're going to discuss this ratio later because this is an important ratio that we want to consider in the gut.
What do you mean by a chimpanzee?
Well, I was given the example that the phylum level, there's different levels of toxicity. Taxonomy. So when you're labeling, when you're naming organisms, it's like, you know, from mammalian, which would go down then to Homo sapien, which would then go down to, to the actual social security number of an individual would be the strain number. So this is the same with bacteria. You're starting way up on the tree. So this is really differentiating between, are they a virus, are they a bacteria, rickettsia, different types higher on the taxonomy level. So I was using human versus chimpanzee just as an example. We're definitely not looking at the difference between humans and chimpanzees in the bacteria, but just to show the relationship between the organisms on the phylum level.
Okay.
And here we're going down to the family level. And it would be like looking, for an example, if we were looking at different races, Africans versus Asians versus Hispanics, so again, if we could look down someone's throat and have all these different species represented by color, you could see this is what it should look like, but this is what your actual sample looks like. So we should have this many of this taupe color and this much salmon, but instead we don't have enough taupe and too much salmon color. So this is just giving you a visual, and here's another way to represent it. We would want most of these organisms on the green stripe, but you see there's a lot that have fallen out. There's some that are too low, some that are too high. And you'll find your preference to which diagrams are helpful and which analogies help you to explain the situation to your patients. Mine tend to be sometimes kind of silly with, the chimpanzees versus the humans. And when there's a takeover, it's like Planet of the Apes, but you just have to tailor your descriptions with your audience. So with that in mind, the Firmicutes versus Bacteroides, Firmicutes, when they're in abundance, that causes a problem for someone to be able to increase their metabolic rate. And we know that those who have slower metabolic rates have harder time losing weight and may be more prone to diabetes. And that's, why we would look at this ratio. So higher Firmicutes is going to be lower metabolic rate. And so each one of these have are important ratios and will have functional purposes to relate to your clients. Here we're at the pathogen. We're looking at that Biophilia wadsworthia, and it gives an example. Escherichia coli was also detected, but it's not out of range. So even though just because it's there doesn't mean it's a problem. But the wadsworthia is a problem. The microbiome is not dealing with this organism as well. And there are some recommendations we can do to help reduce that overgrowth. This is a chart of all of the pathogens that are tested for, and you'll see it's a combination of protozoa, of parasites, viruses, bacteria, and yeast. Now, here's the really interesting part of it. You mentioned that you do diet recommendations. we know that our gut bacteria are very involved with how we break down our food, but there aren't very many tests out there that say if our diet matches our gut bacteria's preferences. We tend to force our preferences onto our gut bacteria. So here's a way to look at it. We have saccharolytic versus proteolytic. Saccharolytic are carbohydrates, proteolytic are proteins. So this is a way to see, is this person's microbiome dealing with their carbohydrates well? So ideally it would be in the green, and this person's sample is in the yellow, so it can be optimized. If we look to see which component is out of range the most in the carbohydrate breakdown, it would be the short chain fatty acid butyrate. And this is a problem because out of the 3 short chain fatty acids, butyrate is the most important. Butyrate is going to be used as a fuel for the mitochondria of the bacteria, as well as the human host. But the mitochondria is important for energy production, for cellular repair, for that energy that drives all the biochemistry of the body. Now, if we go down and look at the proteolytic fermentation, you'll see that it's way out of range. So we'll look for the main culprit behind that, and we'll see that some of the gases, the ammonia and the sulfide are out of balance, but the one that's the most out of balance is the methane production. And of course, this is a problem as far as building up and causing toxicities and pH imbalance in the body, but also it's a problem for the person because usually this person is going to have a lot of flatulence and foul smelling gas, which is a social problem. Now, this is an interesting aspect too, that we look at the psychobiome. So this is a module that's going to look at the neurotransmitters. And you said that you had had some training in neurotransmitters and different tests. And remember, these are the neurotransmitters, hormones, vitamins, gases. All of these are produced by the bacteria. These are not independent analyte studies of what the human is producing, but what these organisms are producing. And they have a very important role in the human biochemistry. And this person here has an issue with GABA production by these organisms. And of course, that's going to make a difference in the gut-brain connection and the emotional side of that has to be considered. The other module here is the Estrabalum. The Estrabalum has to do with how the body's handling estrogen. And if you've done any type of hormone balancing, you'll know that the organisms in our gut can unconjugate the estrogen that is supposed to be removed from our body and cause it to recycle. So these organisms produce an enzyme called beta-glucuronidase. So the more organisms that have more of those enzymes, you're going to have less conjugated estrogen, and that's a problem. So you'll have more recycling of this estrogen. When you start the domino effect of high estrogen dominance, you'll typically see low progesterone, low testosterone, and high cortisol. So that sets up a pattern. And that also is going to affect the emotional stability of the individual you're dealing with. And then here are all of the vitamin biosynthesis. These organisms produce a lot of our B vitamins. And the one that I'm going to use as the example is the vitamin K2. The vitamin K2 is fermented in the gut. Vitamin K1 is made in the liver. And the problem is that a lot of even cardiologists can get the two confused. So it's important to note that the vitamin K2 is the one that has to do with the carboxylation of calcium in the body. So it is removing calcium deposits from soft tissue and putting it back into bone. So because this person's gut microbiome is not producing sufficient amount of vitamin K2, they would be a prime person to put on the supplement megaquinone that we carry. So that gives you an idea how you can get different diet and vitamin and lifestyle recommendations just by looking at these type of results. These are the Keystone strains. And again, each Keystone strain, it tells you what its primary role is. And from this person's, we probably part of their carbohydrate fermentation problem has to do with they don't have a Keystone strain that breaks down cellulose. Their butyrate deficiency has everything to do with the fact that they do not produce a primary organisms that are involved with butyrate production. So this is how you can get some clues, on their microbiome production by what Keystone strains they have and do not have.
Can I ask a question about that?
Yes.
So when you see the tests show that certain ones are not detected versus others are really high, then would you custom design a supplement to leave the appropriate ones that were high out, or add strains in that were not detected to custom balance?
Okay, great question. So what we have learned from our research, and we also do another type of test called a SHINE test, and that stands for synthetic human intestinal modulation experimentation. That's done in Belgium. It's very expensive testing, but it's the only way that you can actually see what products are doing what roles in the gut. So we know that spores colonize in the gut because they have like a shell that gets past the gastric barriers of the stomach acid, the bile salts, and the pancreatic enzymes. And then we also know what they produce when they're in the gut. We also know that other supplements that are not spores, like lactobacillus and bifido probiotics, don't make it to the small intestine. They're digested in the stomach. They work by metabolic signaling, but it's very limited. So we know we can't reseed or replant probiotics into our microbiome. And in fact, even if we could, the microbiome would not accept it, because our microbiome after 2.5 years of age is a mature microbiome, and it closes the door to implantation. What we're given from the environment, from our parents, by 2.5 years old is what we are going to have for the rest of our life, with the exception of being able to influence those by feeding the commensals and out producing the pathogens. And that's what the spores will do. So what we look at with these Keystone strains, we see that the spores are going to increase diversity by 45%. So that's the first thing we want to do, is to help modulate, to bring up the Keystone strains, and then they help keep each other intact into the right ratios. So that's the best way to change the actual organism populations, besides prebiotics that are going to help the Keystone strains thrive, and introducing other key ingredients that we know can change these populations. So yes, part of the answer was customizing the supplements, but typically the organisms that we know are the most effective are going to be the spores, or some that would be introduced in a new technology that would be in an anaerobic delivery, but those have not been launched yet. But I kind of I cue that up, because there are some advances that are going to be coming that we'll announce in later months. Any other questions?
Yes, I just wanted to, I really found it interesting what you just said about it being established and set by the time you're 2 years old. So it's established and set, but yet it can still be changed based on lifestyle and food choices, meaning that the types of strains are already set in there, but they can increase or decrease based on lifestyle, resulting in a more or less healthy diversity. Am I understanding that correctly?
Yes. So the populations are set. Um, Many of them are very low because if there was a C-section, if there were some antibiotics used early on, um, the seed organisms generally are in the appendix, but they never were able to thrive. So we have to work harder for those individuals, but the spores and the total gut restoration, generally we can give those organisms the best chance.
Wonderful. So
Any,
I
go ahead.
think, well, I have a million questions, but I think what I need to do is watch this again on my own and take very extensive notes on what you've taught me. And then from there, do my own research to familiarize myself with the words and exactly what all these little organisms are doing. And I think another way that would be helpful for me to become familiar is to go through the test myself and then go through the total restoration program so I can experience what I'm going to be selling people.
Absolutely. And there are training videos on the site biomefx.com.
Oh,
So that would be a good place.
okay.
And on our website, there are several webinars and clinical grand rounds. And we will be hiring a rep in your area. So there will be an individual to call on you and to coach you through this. And did you have any questions about placing orders? Are you familiar with the site? Do you do virtual clients and want to do patient direct, or do you order things in-house and meet people personally?
I have actually, because of the COVID thing, have um, um, uploaded my entire practice basically to Zoom. So, wait, what was your question again? I'm sorry, ask in-house. What did you ask about in-house?
Yeah, if you're doing virtual, if you go to our website, the patient direct is a way to send a link to your patient so they order from our website, and then they can get the test and they can get the products, or you can order everything into your home or office and then ship it to them. So you've got options, or you can drop ship. It just depends on what works best for you on how you collect for your services. If you like to charge up front, if you like to, have the products dealt with separately, just, there's an option for whatever you want to do.
Okay, so what am I purchasing the test for, wholesale versus retailing it for?
Yeah, so it's $2.99 wholesale, and you can mark that up. I've seen the retail for patient direct. If your patient bought it from us over patient direct would be $3.99. I've seen practitioners mark it up, to $4.50, depending on if you're including your interpretation or some of your consulting fees.
I see. Okay. Um, okay. Is there a place where I can, is this the website, my or BiomeFX, where I can look at those options? So I guess what I don't understand is if the patient is ordering direct from you, would I link some button that onto my website that redirects to your ordering site? And if so, how does my if I settle on the 399 retail, does that factor in for them if I am removed from the ordering process if they order direct from you?
Okay, so I'm going to try to go onto your site. See if I can here.
My site's under construction right now with the products, so the menu bar is a little messy. I'm not going to be done until the end of the week. I'm sorry.
Well, this is the Microbiome Lab site that you can order from. So have you logged into that?
Oh, I see. Is that what we're looking at right now?
I'm trying to get on it. We'll see if I have enough bandwidth. Okay, hang on.
Okay.
Because I don't know if you've ordered any products yet.
No, I haven't. Not at all.
Okay. Okay, so I think it's going to let me on. Try to close some windows here. Okay, so this is your site. And when you go to order, you go to wholesale. This is where you would pick out the products. You would want to get the tests. They're free. The boxes right here, you get a bundle of 4, and you want to, when you order the products, get the kits, and you're not charged for the kit until you register it. So you want to get it in hand, and then when you go to register it, there's a barcode in there, and then you pay for it. So just know you want to get the kits on hand.
I see.
Especially if you want to do a test this month, you will save 20% because they're doing a special. And when you check out, after you register the kit, put BFX20 in the coupon to get that 20% off. But you got to get the kit first. Okay, now for the patient direct, it's over here on the left-hand side, and you would click that. It will have you upload a W-9 because Microbiome Labs is going to be sending you commission checks. So while your patients are ordering on our site, it's going to be accumulating this commission. Once it hits $200, they cut $200 checks. But you have to upload that W-9. You attach your personal code. It's whatever code you want to the link, and that code associates your patients with your account, and that's how they're connected. But it'll coach you through that. After you upload the form, it'll step you through putting your code in there and then sending out this link to other people. And then when you get after you register your lab, your kit, right here will step you through. This is Manage Labs. It's also on the left-hand side. It'll step you through how to register the kit, putting the barcode in, the patient's date of birth or your date of birth if it's for you, and then collect the sample. On that website, BiomeFX, there's a cute little video stepping someone through how to collect their own stool sample. It helps them get over poop fear, but helps them, so they don't make a mistake. And then they mail in the sample. When you get the results back, your results will show up here in the Managed Labs. And then you can, there is a link that you sign up for a consult, and these are experts. All they do day in and day out is interpret BiomeFX tests for practitioners. So when you're ready to explain these test results to a patient, you will have all your questions answered and feel very competent. So that's a free service to you.
And that's, did I understand that correctly, that you say you're including with the results basically a breakdown or explanation on from your end?
Yes. Yes. So there will be the sample report. There's also an interpretation guide on that website and all the recommendations. So you have all of those.
Okay.
I'm just heads
When-
up, I'm supposed to be in a Zoom training a new
Oh, okay.
sales rep. I'm just letting you know here that we can reschedule, or I can keep you in the loop when we get your rep hired, but reach out to me in between time. Let me help you until we get that person in place for you.
Okay. I was curious about, I had spoken with Jeff about possibly applying for the rep position. Have you already filled that role?
That's a good question. I'm not the one who does the hiring, but you know, I will look into that for you. Did you already send your resume in?
No, I didn't. Jeff had said that the next step would have been to speak with you about it, but maybe it's a different person that I should be directed to.
Yeah. So the national sales rep is Tom Hooley, and then his boss is Kendra. So if you want to shoot me a quick email, you can attach your resume. That way it'll expedite. I will send it to those two folks, and we'll see if they've already started interviewing or where the status is.
Okay, wonderful. Thank you so much for your time. I'll rewatch this, and if I have more questions, I'll reach out to schedule again with you if I need if I need anything else.
Perfect. I'll let this convert on my computer. If you don't hear from me in about an hour, add that to that email. It just means that I got distracted.
Okay, thank you so much.
Thank you, Caitlin. Great to meet you.
Bye.
Bye-bye.